Professor Verma is a prominent microbiologist at the Department of Microbiology, where he leads the Bioprocess and Bioenergy Laboratory at CURAJ. He is recognized globally, having been ranked in the top 2% of scientists by Stanford University and named a top scholar in bioprocessing and bioengineering. Professor Verma is a fellow of prestigious organizations, including the National Academy of Biological Sciences and the Biotechnological Research Society of India, and has received numerous accolades such as the Ron Cockcroft Award and the UNESCO Fellowship. With expertise in the microbial diversity, bioremediation, and bioprocess development, Professor Verma has contributed significantly to the field of bioenergy, especially in microwave-assisted biomass pretreatment and biobutanol production, holding several international patents. His prolific academic output includes over 100 research papers, 64 book chapters, and 26 edited books. His work has garnered more than 7,360 citations with an H index of 45 and I index of 109. Congratulations, sir, for the same. Professor Verma has also been involved in postdoctoral research in Europe and Japan and has served as a guest editor for several journals. He is an active reviewer for more than 60 leading journals, solidifying his reputation as a leader in microbial process and engineering. We are so happy, Professor Verma, to have you in our today's session of celebrating our World Fungus Day. Thank you so much for joining us, Professor Verma.
Thank you very much for nice introductions. I hope you are seeing my screen.
Yes, now it is visible. If you can make it full mode, yes, it is visible very clearly, sir.
Yeah. So thank you very much for your nice introduction. I once again thank you, Prof. Deshmukh, to invite me for such a nice forum, which is leading for the fungal mycology being together at a particular level. And apart from this, because as a Professor Joshi talks, because he's a very good friend of mine, so I'm working with the bioprocess development because I'm developing a scale-up process, and I learned a lot of patent on this particular part of it. So I'm working with especially in enzymology and bioprocess development enzyme. So I have particular talk with the Laccases, which is a multifaceted environmental application in a particular fungi, which is we are isolated from the Rajasthan. So you can see the very good campus of us, which is established in 2009. Now it's a wonderful lush green campus. We have it. So it is earlier it was a desert part of India, and you do not have found any vegetations. Now after 14 years of the existence of this campus, it's a wonderful campus and lush green campus around more than 5,000 trees, which is a remarkable development of in the Rajasthan desert part of India. So I will start with my talk with the fungal applications, because fungals are wonderful organism which is still unexplored, because I have one collaborative project with Assam and one collaborative with Rajasthan. So we have explored a lot of fungi and published a lot of data with this. So they are the existence of the fungus is very the diverse ecology niches, and they have very suitable climatic condition, especially in India. That's why we can have to look what the possible application, what the medical uses, what the biological uses of this. And I found very interesting, this diagram which is showing on the slide, which is show the how we can exploit the fungi industrially, and which is biofertilizer, biofuel, medical application. So many applications are there. So I would talk about the enzyme on this particular aspect. So we are working northwest part of Rajasthan, and we are working with the multi-carboxylase, which is laccase, which is a typical laccases, and they be found in both fungi, bacteria, plant. But our interest was the fungi. So we have collected this 3 monsoon session, and we isolate this fungi. We regularly find this thing. We obtain this fungi, and we find very dynamic variety among the laccases, which is the type 1, type 2, type 3, which is designated as a white laccases, blue laccases, and yellow laccases. And the blue laccase, which is very common, which is a species Tapetum versicolor. There are a lot of organisms there. and white laccases, which you have Mycothecium, varivova, and the yellow laccases, Strophoria species are there, which is also available elsewhere. So you can see about the existence of the copper natures. They are dependent upon this. So we have a different sampling site of this in Rajasthan. We have around 7 or 8 sampling sites are there, which is the Dai area, which is Sanghanar painting near Pushkar, Balotra, Jaisol, Srinagar, near Padhapur, Ajmer. We isolate this around 36 fungi, which is look of our interest, whether these fungi especially produce the laccases or not, and what are the diversity of the fungi which is produced in laccases. So we have used the traditional method, which is isolated, and we have sliced with the ethanol, treating with the 3% H2 and sterile dilution. Then we also antibiotic added this to we can get the isolated fungi colonies are there. Among this, we have found 2 interesting fungi, which is Mycothecium varioa and the Stophoria species, which is here we have detailed work and done. And we have also observed that while we observed that we are working with the fungi for the last 20 years, and we find a lot of literature survey, we find a lot of people are working with this cases, and they're using a different methodology for determining the activities are there. So one of the paper we published in Tree Biotech, we are using different method, and we are to say what method is appropriate, because there's a the comparison that this particular strain is producing more activities and in a particular time, but the species are same. So what are those? We observed that different methodology has been used, and the activity methodology has been used, and we have identified those methodologies, and we found that what is the appropriate methodology we can use for this particular purpose to determine the fungi activity. So when we conclude this thing, we use the same different fungus, this fungi, and use this optim media, which is basal media, and one of the methods which I used in 2002. Then we also use the CCD RSM and composite design RSM method, responsible methodology, to determine the activity of this particular, and how many possible variable factors are there which can be increase the variety of the activities are there. So these are the outline of our work which we did. I will explain later stage. So total 36 functions we isolated. Around 22 were not selected because we have not find any laccase activity or significant amount of activities are there. 14 fungus were selected. Among them, we have produced this laccase activities are there. Among the assay, among the 14, we found 3 is the most prominent laccase producing strain which we are looking interest of us, and these are the Mycothium varioa, Strophoria species, and the Pastorella postigua. So these are the fungi. Among the 3, we have used only 2 fungi for further exploration, and we use the shape of conditions, and we find that ABTS method is used for this particular determination, and then we have found that Strophoria species around 2090 unit per liter, and around Mycothecium varioa is around 156 unit per liter produced. So we have used only these 2 fungi for our further studies. So then we have used this particular media, and we determined the variable component, pH, temperature, and this carbon nitrogen source. So where how can we possible combination we can design that can be have a unique what is theoretical yield and what is obtained yield, experimental yield can be done. On the basis of that, we try to found that 1.45 old in increase in the Mycothium varioa, when we see the variation around 30 combinations was used, and we found this increase of 1.45 yield is there. Apart from this, in Stophoria, we can find 2-fold increase in this the media which obtained with the glucose and carbon interaction and pH temperature interactions are there. So after this thing, we can say that in Mycothecium, we, as I said, already 1.45 increase and 2-fold in Stophoria. We also use the purification technique because industrially when we use the purification, large scale productions, and purified enzyme is required. So when we use a traditional method, or that is very expensive method, which has caused the product cost. So we use the ATPase method, which is ATPS with the 30% NA2S for sodium sulfate with the PAX 10%, which we retain the activities almost similar to the conventional techniques, and the molecular weight with the same, which is kilodalton. And the same case of this ATPS, 30% with the sodium sulfate with the 8,000 PAC, 30% 8,000 combination are there. We also optimize this optimization this BCC RSM to obtain the purification procedure, and which is alkaline stable, which is we determine with the conventional and compare with the ATPS method also there. So we have drastically reduced the cost of the purification without losing the yield of the. So we try to use these enzymes for the exploration for the lignin degradation, because one of our laboratory focus is the biofuel production, because biofuel and the cost with the pre-treatment, which is cost around 50% and 25% cost of the enzyme, because the enzyme which is using for the market before the saccharification and simultaneous fermentation when ethanol production is going to place, which 25%. We use the in-house production of the enzymes. That is why we have used this particular Laccases treatment has done, and they have been removed this exposed to the hemicellulose, so they can be attacked susceptible to the gelatinous cellulose enzyme. So, and apart from this, we also use the dye, because this Balotra and Sangana region, we have traditional dye industries are there. We have very different dyes are there, which is we can use for the particular dye degradation, because they are being very toxic in nature, and we also find interesting result with this. So regarding the application, we use this rice straw, wheat straw, sugarcane bagasse, and Lufas and Leca we use for determine this delineation method, and we dried and seized, soaked in sodium acid buffer, and when we treated with the Laccases with 30 degrees centigrade for 12 hours, and we also determine the lignin, calcium lignins, XRD and FTR determinations are going to take place, and you can see that that we have crystalline index are removed, because we have reduced the amorphous samples we exposed. Then lignins are removed with enzyme digestion enhanced, and then maximum in the case of the 4 component products, we use B star and S star. We found the maximum enzyme digestivity and lignin removal are going to take place. That we also determined with the FTR with this thing. We have found this the lignin peaks are there, but 1600 cm, and which is CH2 deformation is going to take place, and relatives maximum in wheat star. And apart from cellular crystallinity, which is also increase the digestibility in wheat star, and the cleavage of the various bondings upon CO, CH, and OH phenolic rings, the maximum in the wheat stars are there. So these are the finding which we can determine with the FTR, and which is very interesting to us to reduce the cost of the product in the biofuel industries. So these are the wheat star, rice star. These are the component which we use for this particular purpose. And we have used this microthecium with the white Laccases, Stohoria with the yellow Laccases. I will detail the characteristic of the what is different with this. So maximum, reductions of the calcium is 64% by this microthecium arboretum, and the kappa number is around 58%, and XRD with crystalline index is which is 34%, because the more crystalline index, which more is amorphous in nature, so that means we can expose to the cellulose, cellulase and gelatinase, so they can be converted into sugar, free sugar, and that can be simultaneously converted into biofuel productions. So in case of this Stophoria, which is 38% and 68% calcium removal are there, and we can see 56% is crystalline index are there. So we can see that these are the with the data with the correlation with the calcium removal and the FTR XRD relations each other. So the white and yellow Laccases behave differently in while removal of the lignin and the things. So we also explore this particular for biodegradation of this paper which we have discussed earlier. We published in 2 very important journal, Bioresearch Technology Reports, and one of the other paper of journal Applied Microbiology, Biocatalysis. Now we also work with this with the dye remediation. This particular 2 type of dyes, which is very important there in the Sangana painting place, which is also nature anthraquinone, azodyes, and basic natures. So these are the sites where which we can we see this which is contamination places are going to take place, and we have been optimized carbon nitrogen source to optimal declaration. They are going to take place. What is the pH and what is the temperature which is going to take place? We determine this declaration efficiency of this, and what is the effective of the removal of the dye is there. So because when we use this in a liquid condition, which is we can use only one time only, so we use the immobilize this enzyme in Scotch Brite and Lithos indica, which is natural carbon source are there, and we have make a cubes of it and grow the fungi in this particular things are there, and we have treated this dyes of 30 degrees Celsius for 10 days, and you can see the flask, which is can say that we have been find very. We also use this in a column reactor, and we also determine this laccase activity among this and protein determination that will take place, and we also regenerate the column because we can up to run 10 cycle because we have to aseptically maintain this column conditions, and we had to regenerate the culture conditions. So after this, we can up to the 10 cycles are going to take place, and we have without losing any efficiency, we up to 10 cycle 10 day we can do this, and we have determined this visual spectroscopy, FTR, LCMS, and phytotoxicity analysis of this particular compound, and you can see that after 24 hours you can completely remove the dyes and both the dyes with the stain, and we have seen this removal dyes and the laccase activity determined that in a column reactor with the Lithos indica and Scotch Brite up to the 10 days, and we can sound the 10 cycle was going to take place. Then same also this is we have also conducted same analysis, which we say the deposition of this particular fungi was there, and they can be entered in the mycelia, in a Lithos indica and the Scotch Brite, and they are going to, that is why they have been continued for long cycles, and we are also able to find out the laccase activity throughout the process. So that is one of the reason why we have been used, and we can see this the XRD when we treated with the treated dyes with the Stoveria and control we have been used where the no treatment are going to place, and compared with this XRD analysis, and we observe this difference in the peaks are there with the similar case in FTA also, which the deformation going to take place certain peaks which is identified with the what peak has been changing. Then we have to identify how much removal going to take place in this particular peaks are there. This is the paper we have published in the biodeclarations. Then we also try to determine the pathway. What pathways we follow? We also use the LCMS acute analysis, and we determine the intermediate products are there, and we determine the pathway because the ALCG, which is going to be oxidized by laccase, and this oxide going to take place, the intermediate product going to take place, and this laccase again oxidized with the presence of oxygen, and they have been stable product going to take place, which is non-toxic in nature, because we also perform the phytotoxicity analysis, and similarly with anthraquinone AR dyes, which is laccase oxidized in presence of oxygen, water release, and this oxide product, which is intermediate product, we again oxygen is going to be place, which we find as another stable intermediate product was there. So we determine this two different pathways are there, which is established and published in biodegradation journal, and both the publication in one second stain we produced in biotechnology reports. So these we established pathway with these two dyes, and we can run up to 10 cycles. So these are the very important finding reasons. These are the culture dependent and enzymatic, which is the green revolution. We can say that because by this the entire fungal I was treated very nice way, and this around 80 to 85%, and 6 cycle we can say 80% that removal will going to take place here. Around 6 cycle or 60% removal going to take place. After that we again regenerate this column, and then again we is going to take place up to 10 cycle. So these are the advantages when we run the column system. So loss of the enzyme is avoided. So we also confirm this the UV spectra XRD after biodegradation of dyes are going to take place. LCMS study also confirmed the proposed biodegradation pathways are going to take place. Phytotoxicity assay also confirmed which is the product, which is intermediate product going to take place, which is less toxic after degradation of the dyes. So overall finding is that we can say that these particular fungi which is produce white laccases, especially Mycotria variva, Astoria yellow laccases, we have established a formula for this, and this is 66 kilodalton. In this case of Stomphoria, is 160 kilodalton was there going to delineation with the wheat straw as and the rice straw was going to be remarkable, which can be enhance the digestibility for the cellulose analysis going to take place. And these are uh, all the three approach we're going to take benefit for the lignin removal and dye degradation. So when we compare with the structural differences on this blue laccases, which is the most common, and the white laccases, which is found in our isolate Mycotria variva, and the yellow laccases, Stoveria species, we find that we had to compare the comparative analysis study going to take place. We found this with LCM analysis, and we observed that when we compared with this bioinformatics analysis, we found this is blue laccase is tomato villosa, and this white Mycotria varivola is equivalent to the bilirubin oxidase. We further verify whether this is the laccases or this thing. We established method, and which is published also, that this is the only the not the bilirubin oxidase. It is a what you say that this is laccases only, and the yellow laccase which is also published tomato versicolor, also the which is also published by our stain, which is Staphylococcus stain. So what is the difference between 605 blue laccase present, because it represents this peaks in copper, and white laccase which is absent, but it is incomplete oxidation, which we see the first graph where we can say the pregnancy, because we comprehensive review has been published by us, which is in bioresource bioprocessing, which is the most downloadable published article of us. So, which is yellow case, which is also incomplete heterogeneity of the glycosylation going to place in yellow cases. So, we also done this XRD analysis, how it is the alpha helix, the beta sheets are going to difference between the yellow case and the blue cases, and we observed this variation in the blue case, white cases, and yellow case of the Stoveria and Mycotria variva, which they compare with the blue cases. So these are the variation which is going to prove that these are the unique characteristic of this particular stain, which is isolated by the digestion. And this also FTA study also given a hint about the differences on this, the structural difference of the blue cases and yellow cases and the blue cases are there. So these are the differences between the amides and white cases are there. So what are the major findings which we conclude in this about this? That white laccases, which is the other study which is mentioned in the table, our its pH is effectively 79. 48 hours we have, we have produced, and this is efficiently remove this lignin and the dyes, and there have been temperature around 30 to 40 degrees centigrade, and molecular the masses are 60 kilodalton, which is equivalent to same. The similarly we Stoveria species, which is also yellow, they have been noted, but in our stain it is 48 hours. It's wide range of pH it was affected, but and 48 hours we stable for this 40 degrees centigrade, and the molecule was similar same. And this is tomato versicolor, which is well-known stain, which is used for the industry. So we have a patent. This particular part is now in Indian patent is published on right now. So these are the things which is we have published in differences, the structural difference in Helion journal. So what are the differences of applications are there? They have been, as I said, in biogas and because digestibility enhanced, so biogas productivity enhanced, biofuel because they are exposed to this amorphous cellulose, hemicellulose going to take place, so they can be used for this thing. We also work with the MFC, which is also the generation electricities are there, and which also help in bioremediation purposes. Desulfurization, biosulfurization also take place. It also uses biosensor also, and they also have a clinical application for decline this remove the laccases by this particular things. We also work with the fiber synthesis, and the defibrication want to take place in juices also there by laccases are there, and food industries they have been used for this particular purpose. And juice defoliation going to take place, enhance the juice, enhance the color stabilities of the juices. So these are the application by which we have talk about this biodigest, biomaterials, fiber synthesis are there, and some of this application what we are doing, and we're trying to establish the process and large scale, and which is be under process. And by this way we can have a utilization agro waste which by the fungi, which is a diverse organism, then treatment of wastewater, which is we can column reactor system can be run, and we can have a green and sustainable approach can be utilized. So these are the lab members, which is we have 4 to 5 PhD passed out, and they have been working. One is in US, and one is in IIT in Law, one is in LPU, and one is now is going to Sweden, and one is working with the industry. And that lot of industry people are contact with us with regarding the application part, especially there's the wine industries, because we have a Ganganagar, their wine industry are there. They have also fermentation problems are there with this thing. We are solving them. So these are we have quite good support from the DBT in tuning project. We also got the DBT budget grant project with the rifle purpose, and we also got the DST grants for this. And first facility with the Suraj, we have a significant amount of this. This is our life science building. We have 3 departments: microbiology on the top floor, second floor we have we are there, and the first floor in biochemistry, and the ground floor biotechnology department are there. We exist in 2011-12. So we have significant appeal in the nationwide and then international wise. So this is all from my side, because we are working with the application part. We are working with the lab to the land level scale process. We have up to the 20 liter. We have fermenter. We are working with this with the industry part of. Thank you all listening to me.
Thank you, Professor Verma sir, for very nice, informative talk on the Laccases enzyme. I think it's a very current practical applications in the biofuel has been going very nicely and very nicely, sir. It's open for the talk, for the question and answer. Please raise your hands for any queries. We can take a couple of questions. Yeah.
Verma sir, very nice talk. Good afternoon.
Good afternoon, sir. How are you?
I'm fine. Thank you, sir.
So, sir, for kind of some of the papers that I had read on Laccases was that for the decolorization also Laccases enzymes are responsible. So it has been very well established, or still been.
It is well established now. It is well established because it is working for last 20 years. Now we are working with the large scale, reactor studies, because we mobilize with the Lefor cylindrica, which is better than the other which is Scotch Brite, and we can run to 10 cycle, which is around 1 liter system are there. But now we try to develop with the 5 liter system with reactor, continuous process there with the industrial water is using for this. So, but only thing is we have to regenerate the column for time to time. That is the thing we have to. Some limitations are there, but we are successfully established the process.
And, sir, many of the white fungi also show very good Laccases activity. So, is it kind of?
Yes. Among them, it's just what you said rightly, because among this, most of them, the vital fungi produce a good amount of Laccases, but to date, there are a lot of Laccases with the blue Laccases only. And these are the fungi which is isolated. These are the showing the white Laccases and the yellow Laccases, which is very rare studies are going to place, because one of our collaborators in Russia, they are also working this thing. That's why they visited our university last year, and we are collaborating with them in Russia. Because they are working on the white Laccases in Russia. They have been very good mushroom. As a talk which is saw by Professor Joshi, they have been working with the mushroom in Russia. They have eatable mushrooms are working on this. So they have been large scale cultivations are there with going to place.
Fine. Sir, there was a question regarding why there is on 4th day there is a regeneration, as you have mentioned in your slides.
See, because what is there initially we have mentioned is that how much concentration dye is there. Okay, So if it is that we are using a high concentration because industry has a high load in there, so we have to use, if you want to retain the column, that's why we have to 4-day we have to retain. If you using a high concentration of dyes are there, or pH is also variable sometimes because it is the industrial effluents are there. So we had to regenerate the column. If there is the mushroom, this fungus will not be survive there. So how can we can remove this thing? So that's why regeneration going to, it is only for 24 hours regeneration going to take place. After regeneration, we are again for 3 days run the system, and then we run for 10 days like this system.
So we have a question by Mr. Jackson. Jackson, please go ahead with your question.
Sir, it was a very wonderful talk, and to be very honest, today I only know about the different type of lacquers, white, yellow, and blue, and all those things.
Yeah.
Sir, I'm a young researcher. I'm very interested in microbial enzymes, and I have read a little bit about laccases. So, sir, right now we have a burning situation on plastic and microplastic. Sir, do you?
Yes.
I have read literature on lacquers being able to biodegrade microplastic and all. But now, sir, can you
Yes.
which type of lacquers should I focus on if I have to work on this biodegradation part?
See, as per my understanding till now, I was earlier with my PhD with the blue laccases, but now with the white laccases, which we recently patented, and we also started to, because a lot of people are working on microplastic, which is also laccases also helping to remove. So my understanding with this white laccases are microthecium is very good compared to the yellow laccases. So I suggest, because if you go man the paper, which is bioprocessing, which is open access, so you can download this paper, you can find a lot of citations of this paper which was published by us in 2017. More than 500 citations are there. So you can download this paper and you can find a lot of limitations, applications part have been discussed in this particular review article.
Thank you so much, sir. Thank you.
I think, sir, if you can stop sharing the screen, it will be helpful for us. Yeah, I think since there are no more questions, so I think I'll, on behalf of Association of Fungal Biologists and Micro India, I thank Professor Verma and Professor Joshi for celebrating World Fungus Day with us. As Professor Bhatt rightly mentioned, and we congratulate to all on this occasion. I'll just ask the President of Fungal Association of Fungal Biologists, Dr. Sunil Deshmukh sir, to have say a few words on this occasion and the concluding session. Yes.
Actually, I would only thank both of the speakers. They have readily agreed to give a talk on respective topics, and I was aware that both are working on this higher fungi, so I kept both the talks together. That was the only idea. Thank you very much for giving a talk.
Thank you, sir.
Thank you, sir. Thank you so much for joining us with all, and thank you all the participants to joining with us for the day. Thank you so much. Thank you.
Yeah, thank you. Professor, I'll call you.
Yeah, I will talk to you later.
Thank you so much.
Thank you, sir.
Have a good day to all.
Thank you, sir.